A full cycle of development of Drosophila from fertilized egg through 3 larval stages and a pupal stage to the adult takes 10 to 12 days at 25 degrees Celsius. At fertilization, the 2 parental nuclei migrate toward the center of the cell and fuse to form a diploid zygote nucleus. This nucleus undergoes several mitotic divisions without cytokinesis, producing a structure called a syncytium. Most of the nuclei migrate to the periphery of the egg and eventually lead to somatic cells, but a few migrate to the polar cytoplasm, where they become the precursors of germline cells. Membranes form around the nuclei, producing a cellular blastoderm. Subsequent development of body structures involves a well-ordered sequence of developmentally programmed events under strict genetic control. Genes that are involved in the regulation and direction of development are revealed by mutations that affect development. There are 3 major classes of these genes: maternal effect genes, which are expressed by the mother during oogenesis and establish the anterior-posterior and dorsal-ventral polarities of the embryo; segmentation genes, which determine the segments within the embryo and adult fly; and homeotic genes, which specify the identity of each segment with respect to the body part that will develop at metamorphosis. The bicoid BCD gene is an example of a maternal effect gene. The mother transcribes the bicoid gene during oogenesis, and the resulting mRNA becomes localized near the anterior pole of the egg. After fertilization, translation of the mRNA produces the bicoid protein, which diffuses to form a concentration gradient from the anterior end of the fertilized egg to the posterior end. Bicoid protein acts as a transcription factor that both activates and represses genes along the anterior-posterior axis of the embryo. In addition, bicoid represses translation of the mRNA of the caudal CAD gene, leading to a gradient of caudal proteins with the highest concentration at the posterior end of the egg. The caudal protein is involved later in the segmentation phase of development to activate genes required for the formation of posterior structures. The segmentation genes are divided into 3 groups: the gap genes, the pair rule genes, and the segment polarity genes. Mutations in the gap genes, which include quadruple, hunchback, giant, and tailless, result in the deletion of more than 2 adjacent segments. Mutations in the pair rule genes, which include hairy, even-skipped, runt, and Fushi-Terazu, result in the deletion of the same part of the pattern in every other segment. Mutations in the segment polarity genes, which include engrailed, hedgehog, armadillo, and gooseberry, result in portions of segments being replaced by mirror images of adjacent half segments. Homeotic genes code for transcription regulators. The genes share a 180 base pair sequence called the homeobox, which corresponds to a 60 amino acid DNA binding segment of the protein called the homeodomain. The homeodomain containing proteins regulate the expression of genes that are involved in the creation of entire systems of body parts or organ systems. Many mutations in homeotic genes are lethal. However, some non-lethal homeotic gene mutations are known. The effect of these mutations is to alter the identity of particular segments of the fly body, changing them into copies of other segments. For example, a specific set of mutations in the cluster of homeotic genes known as the bithorax complex, BXC, leads to an adult fly that has 2 pairs of wings, one on segment thorax 2, T2, where wings are normally found, and one on thorax 3, T3, which normally would contain a pair of rudimentary wings called halteres.